So now we'll move to our first spotlight session of the conference to discuss really an exciting initiative that is transforming the way we conduct research and treat cancers. Dr. Newcomer alluded to it and applauded it just earlier, so perfect timing for that. We're very fortunate this morning to hear from Dr. Roy Herbst, who will present the Lung Master Protocol, otherwise known as LungMap. In addition to his day job where he serves as the Ensign Professor of Medicine and Chief of Medical Oncology at the Yale Cancer Center, Dr. Herbst is also one of the leaders of this innovative way to get drugs to patients faster. In addition, Dr. Herbst is very much involved in the AACR Science Policy and Government Affairs Committee, as well as our Regulatory Science and Policy Subcommittee. He's a leader of our Tobacco and Cancer Task Force. So we have all kinds of interactions with Dr. Herbst, and we're just thrilled that he's here to join us this morning.
Well, thanks, John. It's really a pleasure to be here today and to see so many of you and to talk about this trial as an example of one of the ways we're trying to pull it all together, to bring the drugs to the patients at the right time throughout the entire country. And you'll see how the Master Protocol really has tried to do just that. I have a few slides that you should see on the screen, but mostly I'll give you my comments. You know, this all came about, because of the need to help patients, to get drugs to patients, to get drugs approved. We realized that our clinical trial system was broken. You heard that from the panel, the fact that many patients don't even enroll on protocols. They're not available. Patients can't get in because of eligibility criteria. And there was a report from the IOM, and looking around the room, I see many people who are authors on this report that really set out to say, how can we do better? How can we do more impactful trials? Trials where when they finish accruing, the drug is still relevant. Trials where we can find a drug for those 5% of patients that have a molecular abnormality. We have to screen 100 patients. We can't do that with one-off trials. We need one trial where we can screen many patients that can get on the study. That's great for patients. It's also great for getting drugs approved in a faster way. So that was all well and good, but then there was the question of how do we implement this report? In an era where research funds are even less than they were when the report was written, the groups have all come together in a very nice network. So I think you'll see the Master Protocol is designed to try to implement the scientific questions that were raised in this report. So we got together, and there were several groups that formed task forces to look at this. I was very fortunate to get a call one day from Ellen Siegel from the Friends of Cancer Research to prepare a white paper for the Brookings meeting, this being more than 2 years ago, to ask how could we do a master protocol. And she said, well, why don't we focus on lung cancer? And I said, that's great. At the same time, the NCI steering committee was having similar discussions, and as you'll see, we pooled all the efforts, and we're running this trial through the NCI mechanism. In order to find drugs for patients, there are a number of ways to do it. I show the umbrella and the basket. We were just discussing this at our meeting yesterday at Yale, explaining this to our staff. Nowadays, as we have all the molecular studies and we know so much about a patient's tumor, we have to find the right drug. A basket trial would be trials, many different tumor types perhaps, where you have a molecular abnormality, and now we have trials that will accept patients no matter what the tumor type for that abnormality. And many centers are holding personalized tumor boards once a week, adding that to the lung, breast, and colon tumor board, trying to match the right drug to the patient. Those are usually earlier phase trials. What we wanted to develop was an umbrella trial, a trial for one type of lung cancer. You'll see it's refractory, second-line squamous cell lung cancer, and try to find a way to match drugs to patients and to see if this could go all the way through to phase 3. So we've set out, and I represent a large group, many people working across many public-private partnership associations, and I'll show you some of that as I speak. Well, the first thing was, what do we target? So we wanted to target something that was doable, that was unmet in its need, that was based on new science. And what I show here is in the last 5 years, the TCGA project has resulted in a great deal of information about squamous cell lung cancer. Lung cancer is the #1 cause of cancer death, nearly 200,000 deaths a year in the United States. There have been amazing advances in the last 10, 15 years. EGFR inhibitors, ALK inhibitors, immune therapy is on the rise. But I'll tell you that many of the small molecule inhibitors, they work mostly for the patients who have not smoked, for the adenocarcinoma patients. And all of those patients ultimately, by the way, become resistant. So here we said we'll target really the unmet need of squamous lung cancer. Almost all of these patients have smoked. They're the other 80% or more, the patients who are resistant. And we knew that we had some leads because we knew that there were some mutations. And you can see on this slide, for those who can't see it, I'll just describe. PI3 kinase mutations, 9%. But you can't do that as a single-arm trial. You can't do that at one site. This is where we need the cooperation to bring the drugs to the patients, the companies working with the academic centers, with the NCI. So here were the major goals, and are the major goals. This is a very much ongoing effort. The trial's running right now at close to 250 centers in the United States. We wanted a mechanism for genomic screening of large homogeneous patient populations. One-stop shopping. You come into the lung clinic, in the community, at an academic center, no matter where you go, you get profiled, and that profile then gives you the key to get one of 5 different drugs. We wanted to accrue and assign it simultaneously to multiple sub-studies and using this sort of master protocol design to compare a new targeted therapy to a standard of care. So the thing that makes this different than some of the other earlier iterations, the BATTLE trial, the I-SPY trial, is this is comparing to standard of care. This has a phase 2 interim analysis, and then it can go all the way to phase 3. And one important thing to mention is much of the push for this actually came from the FDA. Through informal discussions with Drs. Woodcock, Pazder, and others, they actually urged the groups and challenged us to do this because the ultimate goal, as you can see at the bottom, is to identify and quickly lead to approval of safe and effective treatments for patients with lung cancer based on a biomarker target. So here basically is the schema of how it works. A patient will register and come in and sign consent. So someone comes in, they've failed therapy for their squamous lung cancer, and sadly that's most patients that will occur. They usually have had a platinum-based therapy, very small improvement in survival, if any. And then what happens is they come in and we collect the tumor. If the patient has tissue, that's great. If they don't have tissue, the trial pays for the biopsy. This is bringing biopsies to patients, and then it gets profiled. And you know what? We're going to talk a lot about cost and value and how things are paid for. It's all paid for in this trial. Why? Because it's a public-private partnership, a $170 million partnership with 3/4 or more of that money coming from the private part using the NCI and the cooperative group structure. It really is working well to help patients. Then the patient is assigned to one of 5 arms. I'll show you an example of that, how that works. And they either get the investigational targeted therapy on that arm or they get a standard of care. It is a phase 3 trial. It is a randomized trial, but it's helping patients in real time, and we're learning so that more patients can be helped in the future if these drugs, in fact, do become approved. Now, some of these drugs might not work, and we have early stopping rules, and then it's a modular design so that we can add or delete an arm of the trial. So that's shown here, and you can basically see that the first iteration of this trial, which started in June, we're about 3+ months into it now, has biomarker profiling. We're using a Foundation Medicine platform. Patients receive the biomarker result with their physician, and there are 4 arms of targeted therapy, A, B, C, and D, where they can get assigned to based on their specific biomarker status. And within those arms, they either get the targeted therapy, A, B, C, or D are shown here, or a chemotherapy control. Now, we realize that not every patient's going to have a biomarker. One of the biggest challenges of this effort, I'll tell you, is finding good drugs that are paired with good biomarkers. So the other 50% are getting immunotherapy, and I can tell you that immunotherapy is changing the way we look at lung cancer right now. It works in a large number of patients. We still need to find biomarkers, but here we're not using a biomarker to randomize patients to immunotherapy versus control. But you know what? We're looking back, and the discovery that will come from this trial, the DNA sequencing, the ability to figure out which biomarkers do correlate with immunotherapy response, will be very important. So we set out to do this trial, and there was amazing interest. Ellen and the friends, the NCI steering committee, we had multiple meetings in Washington. Many of you were there. And I can tell you almost every company came to us with a drug or a target. Over the course of last summer, we held 15 different meetings. We put together a committee that included all the cooperative group lung shares, members that had expertise in different drug classes, industry leaders who had been with companies but now were consulting so that they had at least a less conflict of interest. And we actually reviewed drugs that were proposed by different groups, and we picked 5 drugs for the first iteration of this trial. For the sake of time, it's not really that important, but you'll just see that AstraZeneca is involved with their drug. This is a drug that targets fibroblast growth factor receptor. Genentech with their drug, which is a beta-sparing PI3 kinase inhibitor. MedImmune with their PD-L1 antibody. Pfizer with their CDK4/6 palbociclib, and Amgen with an antibody against HGF, hepatocyte growth factor, rocatinlimab. And there are many other companies that we're talking to now. We're already planning for the next arms. Again, some of these will, hopefully all of these will succeed. If they don't, we'll learn and we'll move on. Now, the profiling. One of the main things that this is doing is it's bringing profiling to the community. I don't know the exact figures. Perhaps we'll hear it during the course of the day. But I can tell you from my experience at Yale, prior to that at MD Anderson, I stay in business because people come without profiling. So I'm able to then see them in my clinic, get the tissue, and profile them. So this type of trial is bringing profiling to patients throughout the country. And you can see here that we're using a foundation medicine platform, very similar to their commercial platform, though it's a slightly different type of report, looking at about 250 different genes. Vince Miller, who's leading this effort at Foundation, sent me an example this morning, anonymous, of course, but here's an example of what the physician would get. Actually, first it goes to SWOG, the patient's assigned to the arm, but information's all made available to the physicians and the patients. And the reason for that is we had meetings such as this as we were developing it. We really wanted to be a grassroots trial where we talked to the patients and the caregivers, and they said, we want to have all the information. So the patient gets a report like this that shows the abnormalities, but also has access to the full Foundation Medicine report, should they want it. Patients have the full choice to do what they want. So here's basically, in summary, the LungMap trial. It's a public-private partnership. It's an effort among many groups. It's working very well. We're now open around the country. You can see here on this next slide that we have about 200 sites open now. Our plans are to open at about 500 sites. Very good news. We've had interest from Canada. So right now we're working through to get the NCI Canada involved. I think that's going to be a very big advance because they've been very good in the past at accruing patients to cooperative group studies, where we've been approached by private groups. I won't say which ones since it's not final yet, but 2 groups that typically don't participate in cooperative group trials are now joining cooperative groups so that they can be involved in the trial. And Jeff Abrams has helped us very much with that. And we also are continuing to expand our reach, really wanting to have this throughout the community. Our approach here, slightly different from the iSpy approach, is to try to bring this to as many sites as possible. Now, accrual is always an issue. We have great resources, we have great expectations, so our team is working quite hard. In fact, I have several meetings today to try to keep the accrual of this trial on a very high track. We've also had interest from Congress, and I think that's where I want to end before I take a few questions. So I was asked to talk before the 21st Century Cures Committee. I know several others in this room have as well. And really, we use this as an example to say, this is what we need to do to help patients. We need to find trials that can bring drugs to patients in a mechanism where they can get analyzed, they can be approved by this mechanism, or a decision can be made to move on to the next drug. So the development of biomarkers is an outcome of this trial, and now there are other groups in colon and breast cancer in the US and throughout the world who are doing the same design. We have to develop a better diagnostic test early on for some of these markers. The partnership, I think, is huge. The fact that we have the NCI working with the FDA, working with Friends of Cancer Research, working with the cooperative groups. In this case, SWOG is leading, but in a similar trial, another group might lead. Working with the patient advocates, working with the physicians throughout the country. It really is wonderful. And then, of course, the companies who have figured out that by coming to this mechanism, they can do their trials hopefully faster, and we're sharing the data, as you heard earlier. Now, there are some firewalls for companies for specific things that involve their proprietary interests, but really everyone's working together to get their drugs tested and hopefully approved a bit faster. So that's really all I want to say. It's again, I represent a large group, and it really is, I think, a way of the future, and the fact that we can channel private money into the public system, use an infrastructure that wasn't really that broken, it just needed some correction, because the cooperative groups really do get out to the sites, and now to really keep this trial on track, analyze the data in real time, both to understand if the drugs do work or don't work, but also to discover new markers through the process as well. And the nice thing about this is many people are now already thinking about using the tissue that's obtained here to discover new markers or new combinations, because as we heard in the last session, it's going to be novel combinations that might be the way of the future. And with that, I'll stop and thank you very much.
You covered it all, Dr. Herbst.
Well, I'm happy to talk informally at the break.
Fantastic. All right, thank you so much, Dr. Herbst. Oh, actually, we do have one question. Okay.
Given the limited benefit of chemotherapy as first-line treatment of non-small cell lung cancer, squamous carcinoma particularly, what was the thinking about using this in second line as opposed to using this protocol for first-line treatment?
Well, yeah, I think you make a good point. However, you know, the evidence for these agents at this point was not strong enough to bring it front line. As little benefit as chemotherapy provides, and I'm very much a proponent of the new molecular therapies, there is some benefit to chemotherapy in that setting. What we've had to do, and this has been the hardest part of this trial, finding drugs, even though you have the target, but finding drugs for which there's enough evidence that there's a response rate or activity in that molecular subset. And one thing that I didn't say for the sake of time is it's always not clear. There are many, it's not just PI3 kinase mutation. There are many different mutations. So the complexity of this is quite large. So we figured that to bring this to front line until we actually tested this and reduce the variables a little bit, which specific mutations respond? Are we seeing a response rate? What's the toxicity? It was just too soon. But you're right. The best thing to do would ultimately be to find drugs, get them approved perhaps in this setting, and then bring them up as early as possible.
Thank you. On the trial design, and specifically the issue of, is it a clinically meaningful difference between the targeted therapy and the standard of care? So it's easy to define futility, it just doesn't work. But when you did your sample size calculation for the various 5 arms, you got a lot of patients, how much better did the targeted therapy have to be considered a success?
Right. Well, I know a bit about this. You know, I was on the ASCO Meaningful Outcomes Committee, and we basically, in order to make it past the phase 2 to phase 3, looking at a PFS endpoint, we wanted to have a 2-fold improvement in progression-free survival. In fact, that wasn't good enough for some of the companies, because as I know there are people here from companies, some of their internal standards want an even higher bar, because going to phase 3 is where it gets much more expensive, right? So 2 of the groups chose a 2.5 fold increase. That's Plan B. Our hazard ratio is 0.7. You know, we can argue what hazard ratio is the right one, but we felt that would be a meaningful benefit. You're right, though. For a targeted therapy, you might want to see even better, but many of these agents might not be. It might not be the same effect you see with an EGFR inhibitor in an EGFR mutated patient, but we're hoping for better. You're right. You'd like to see home runs if you have the right drug for the right target. But the problem is we're probably going to need to start moving to combinations, and that could get a little bit more complicated.